All right, let's dive in, fellow microbiologists. Ready for a deep dive into antibiotics, specifically the ones that target those bacterial membranes.
Always a hot topic.
Yeah, definitely. And we've got a fantastic resource to guide us today. We'll be looking at Antibiotics, Challenges, Mechanisms, Opportunities by Walsh and Wincevich.
A really great book, I have to say.
It is. It's packed with insights. And, as medical microbiologists, I think we're always looking for ways to stay ahead of the curve, especially when it comes to antibiotic resistance.
Absolutely. Knowledge is power, right? Especially in our field.
Right. So let's jump right in. Now, resistance is a huge problem, obviously, but what I find fascinating is that nature has already figured out ways to mess with those bacterial membranes.
Oh, yeah, for sure. Just think about those antimicrobial peptides, the AMPs, the defensins our immune systems use.
Oh, yeah, defensins. We see their effects all the time. But I have to admit, sometimes I forget just how they work at that molecular level.
Well, think of it like this. There's this model called the carpet model. Imagine, right, these AMPs kind of gathering on the bacterial membrane surface, like they're rolling out a carpet.
Ah, I see. A molecular carpet bomb.
Exactly. And once enough of them are there, boom, the membrane gets all unstable, starts leaking, and the cell, well,
It's toast.
pretty much.
But here's what I find really cool. Not all AMPs are about just straight up killing the bacteria.
Yeah, interesting. You're thinking about something specific.
Yeah, like human defensin 6. Instead of destroying the bacteria outright, it actually forms these nanonets that trap them.
Wow, that's incredible. Almost like a bacterial jail.
Right. And as a medical microbiologist, that makes you think, what if we could design therapies that, disarm the bacteria without just killing them? Maybe that could help with the whole resistance problem.
That's a really thought-provoking idea. Shifting gears a bit, have you ever thought much about lantibiotics?
Antibiotics, yeah, definitely. Those are the peptides that hit both the membrane and lipid tube, right?
Bingo. And as microbiologists, we know how vital lipid 2 is for those bacterial cell walls. So, targeting it is a pretty effective way to stop them in their tracks.
For sure. And there's that classic example, nisin, the one they use to preserve food.
Oh, yeah, nisin. But there are some others out there, too, like microbisporicin. It's had some interesting modifications that make it even more potent.
Modifications, that's intriguing. So we could potentially engineer these peptides to be even more effective.
Precisely. Imagine tweaking their structure to target very specific bacteria or to get around those pesky resistance mechanisms.
Now, that would be a powerful tool to have in our arsenal. All right, let's switch gears again. This time to a protein that takes a totally different approach. Have you heard of calprotectin?
Calprotectin, yeah, it's a bit of a different beast.
Exactly. It doesn't attack the membrane directly.
Nope. Instead, it's like it basically starves the bacteria to death.
Starves them. How so?
Well, it binds to all those metals that bacteria need, like manganese and iron, makes them unavailable.
Ah, so it's like cutting off their supply lines.
Exactly. And you know how crucial those metal ions are for bacterial growth? Pretty clever, isn't it?
Super clever. Makes you wonder if we've fully explored the potential of this, metal deprivation strategy.
Good point. And what's interesting? Clinically, we actually use calprotectin levels to assess inflammatory bowel disease.
Really? So it's not just some theoretical concept. It's actually got real-world applications.
Absolutely. And speaking of real-world applications, let's shift gears one more time, this time to bacterial lipopeptides. The ones bacteria themselves produce.
Oh, yeah. And some of those have turned out to be pretty valuable antibiotics, haven't they?
For sure. A classic example is daptomycin. That workhorse against gram-positive bacteria, especially MRSA.
Daptomycin. I always found it fascinating how it needs that calcium spark to really get going.
It does. It's programmed to work specifically where those bacteria are trying to invade.
Exactly. So it binds to calcium, and then boom, it's in the membrane, disrupting everything.
Pretty much. But of course, there's always that shadow lurking, resistance.
Of course. And some bacteria have evolved ways to change their membranes, basically repelling daptomycin.
Like they put up a keep out sign.
Right. But that actually brings us to a really interesting point, the return of polymyxins.
Polymyxins, yeah, those older antibiotics that we kind of put on the back burner for a while because of those toxicity worries.
Yeah, exactly.
Yeah.
But now they're making a comeback, especially with those multi-drug resistant gram-negative bacteria.
Yep. And that's because they target something very specific, the lipopolysaccharide or LPS layer, that outer membrane of the gram-negative.
So they basically disrupt that outer layer, allowing the antibiotic to get in.
Exactly. It's all about that unique target.
So we've got polymyxins for the gram-negatives, daptomycin working mostly on the gram-positives. As medical microbiologists, we really need to be on top of those differences to pick the right antibiotic, right?
Oh, absolutely. It's antimicrobial stewardship 101.
For sure. But speaking of innovative approaches, have you heard much about the second-generation lipoglycopeptides?
Oh, yeah, those are interesting. They're like a combination
You're
approach.
talking about telavancin, ordovancin, dalbavancin, that group, right?
Exactly. And what makes them so cool is that they bind to lipid 2, just like vancomycin, but then they also go after the membrane itself.
Ah, so it's a double whammy.
Precisely. Gives them that extra punch, and it also helps them go after bacteria hiding in biofilms.
Biofilms, those are always a nightmare to treat. So having something that can actually penetrate them, that's a game changer.
Absolutely. And the key takeaway here, I think, is that combining mechanisms, like what we see with these second-generation lipoglycopeptides, that could be a key strategy for overcoming resistance.
Hit them with multiple attacks, make it harder for them to evolve resistance.
Exactly. A multi-pronged approach.
All right, before we wrap up this part of our deep dive, I want to put your knowledge to the test, a little exam-style pop quiz. Are you ready?
Bring it on. Let's see what we've learned so far.
All right, first question. What makes daptomycin's mechanism of action so unique compared to other lipopeptides? Think carefully. We'll discuss the answer in a bit.
Okay, ready for question number 2?
Hit me with it.
All right. Describe 2 ways bacteria can develop resistance to those membrane-targeting antibiotics we've been discussing.
2 ways. Give me a sec to think.
Take your time, no pressure.
Okay, ready for the next one.
All right, question number 3. Why are polymyxins so good at tackling those gram-negative bacteria while daptomycin isn't really effective against them? It really makes you think about those structural differences, doesn't it?
It does. Okay, how many more questions do we have?
Just 2 more, and then you can breathe easy.
All right, bring it on.
Here's question 4. What's the big advantage of those dual-action antibiotics like telavancin? Think back to how they actually work.
Okay, got it.
And last but not least, question 5. This one's a bit different. How does calprotectin actually work to fight those bacteria? Remember, it's not about messing with the membrane directly.
Okay, that was a good one. Think I got my answers.
Pens down. Let's see how you did.
Time for the big reveal.
All right, for question 1, the answer is daptomycin's reliance on calcium. Unlike other lipopeptides, it needs to bind to those calcium ions to create this complex that can actually insert itself into the bacterial membrane and cause all that disruption. And as medical microbiologists, we know that calcium is just everywhere in the human body, especially at those infection sites. So daptomycin is really effective in those environments.
It's not just being a lipopeptide, it's that calcium trigger that makes it special. What about those 2 ways bacteria can develop resistance to these membrane-targeting antibiotics?
One way is by changing their membranes. They can adjust the types of phospholipids they have, or they can even add positive charges to their surface to push away those positively charged antibiotics like daptomycin.
It's like they're building a wall to keep the antibiotics out.
Exactly. And the other way they can develop resistance,
Tell me.
they can make these things called afflux pumps.
Flux pumps, what are those?
Well, they're basically pumps that actively remove the antibiotic from the cell.
So it's like they're throwing the antibiotic right back out.
Exactly. Prevents the antibiotic from reaching that critical concentration it needs to do its job. Bacteria are crafty, aren't they?
So sneaky. All right, on to question 3. Polymyxins versus daptomycin. Why do polymyxins work against gram-negatives when daptomycin doesn't?
It all comes down to what they're actually targeting. Polymyxins are all about that LPS layer, that special feature you find on the outer membrane of gram-negative bacteria. So they disrupt that membrane, creating a way for the antibiotic to get inside. But daptomycin, well, it mainly targets the cytoplasmic membrane, which is much easier to get to in gram-positive bacteria because they don't have that extra outer membrane.
So it's all about accessibility. Makes sense.
Yeah.
Now, what about the advantage of those dual-action antibiotics like telavancin?
The dual action mechanism is really a big deal. By targeting both lipid 2 and the membrane itself, these antibiotics are much more effective, and bacteria have a harder time developing resistance to them. It's like throwing a 1-2 punch, you know?
Makes it much harder for the bacteria to dodge the attack. And finally, how does calprotectin work its magic?
Calprotectin basically kidnaps essential metals like manganese and iron. By binding to them, it effectively starves the bacteria, preventing them from growing and spreading.
Such a unique strategy. It really highlights how many different ways there are to target bacteria.
Absolutely. And as medical microbiologists, understanding these diverse mechanisms is crucial for developing new strategies to fight those bacterial infections.
So we've covered a lot of ground today, haven't we? All sorts of antibiotics that target bacterial membranes. It's amazing how many different strategies there are.
It really is. From natural peptides to these complex lipoglycopeptides, it's pretty remarkable.
And as medical microbiologists, it's easy to get caught up in the everyday work, diagnosing, treating infections. But sometimes it's good to step back and just appreciate the complexity of these mechanisms.
Absolutely. Keeps the brain engaged, right?
It does.
Yeah.
And it reminds us why we got into this field in the first place. All that intricate detail, fascinating.
Totally agree. And it really underscores how important it is to stay curious, to keep researching, keep exploring, because the threat of resistance is always there.
Oh, for sure. We can't let our guard down. We have to stay ahead of the game. And, like we talked about earlier, nature has already come up with some pretty ingenious solutions.
Yeah.
Like that defensin 6 with its little capture trick.
That one's a real head scratcher. It makes you wonder what other secrets are hiding out there in the natural world, just waiting to be discovered.
Exactly.
Yeah.
And it also highlights how important it is to collaborate with other scientists, chemists, bioengineers, all those folks. Because by working together, we can take those natural strategies and turn them into new therapies.
Couldn't agree more. Teamwork makes the dream work, as they say.
For sure. And for our listeners who want to really dig into this topic, I highly recommend that chapter in Antibiotics, Challenges, Mechanisms, Opportunities by Walsh and Wensvich. It's a treasure trove of information.
Absolutely. A must-read for any medical microbiologist who's serious about their craft.
Couldn't have said it better myself. So as we wrap up this deep dive, I want to leave you with a final thought. We've seen how diverse these mechanisms can be, and we've talked about the challenge of resistance. But the question remains, what's next? What other innovative approaches are out there? What will be the next breakthrough in our fight against bacterial infections? Something to ponder as you go back to your labs and your research.
Lots to think about and lots to discover. Thanks for joining us on this deep dive.
It's been a pleasure. Until next time, stay curious, fellow microbiologists.